All right, folks, let's start. Let's finish this so that we can just talk about a little bit for the upcoming exam, a little bit of review. I have posted the review in your. You will see that there is a clicker question module at the very bottom, and today's clicker question, I will update that. Then you will have all the clicker questions there, and then you will also do have the review. Again, the review means that it's a comprehensive list of the material that we covered. It is not really a study guide. So it's all in there. So what we are trying to do today, I will just highlight some of the areas that I have seen that historically people struggle with, and we're going to talk about. But let's finish this one. All right, so we finished this one, which is the TIPS and the end-binding protein. And right here, we are looking at the third group of protein, which are the proteins that regulate the microtubule disassembly. And remember that microtubule automatically disassemble when the GTP is replaced by the GDP in the beta tubulin and towards the minus end. But the plus end also can disassemble. And as you know, that the plus end will be the beta tubulins are still bind with the GTP. So it is not favorable, and the critical concentration in the cell naturally doesn't favor the depolymerization at the plus end. So to do that, there is a group of protein. One of them is the kinesin 13. It's very unique. That's why you definitely want to underline. This is a motor protein family, but it is not a motor protein. The reason that it is called kinesin 13 because it does have a homology with the other motor proteins, the sequence homologies. That's why they're grouped into that. But this doesn't carry cargo. It just bends to these dimers of alpha and beta tubulin and bends the plus end, and it does that in an ATP dependent fashion. So this disassembly needs ATP, and binding this will bend the plus end, and this destabilizes the microtubule and it will disassemble. So here one of the questions usually can be asked, see the difference between that. This kinesin 13, this is one that we talked about. The next one is a similar, which is a stathmin. The stathmin enhances the GTP hydrolysis, so the mechanism is very different. This is a comparative question that can be asked, that here what it's trying to do, that here the kinesin 13 with the help of an ATP, hydrolyzed to ADP, this is for its own purpose. It binds to the microtubule and just bends it, and that causes the disassembly. It does not replace the GTP to GDP like the other one. On the other hand, the stathmin enhances the GTP hydrolysis. This is very specific. I will not ask that this binds to 2 molecules, this binds to 4 molecules. Those are not very specific. Those are just so do not count on those numbers. Just remember that it also binds and bends the plus end and causes the depolymerization at the plus end. And this enhances the GTP hydrolysis. Make sure that you understand that this GTP is not required by the stathmin. This is the GTP hydrolysis of the beta tubulin. So the beta tubulin, which is bind with the GTP, and automatically beta tubulin by itself, when it treadmills towards the minus end, it will hydrolyze the GTP to GDP by itself and get depolymerized. Same way that actin from ATP to ADP goes towards the minus end and depolymerized. But the only function for the stathmin is to enhance that hydrolysis process so that they disassemble. And also it physically puts a bend, and that destabilizes it. So these are some of the two of the proteins that are responsible for the disassembly at the plus end. So after that, we are looking at that this is another one that we have seen this picture, and your natural question could be that why you need the disassembly at the plus end. This is the reason that you need the disassembly at the plus end. Remember that when a cell is dividing, particularly in a mitosis, your microtubule organization center is on each pole, centrioles divided. Now you have all these asters which are going towards the middle of the cell or towards the periphery. These are plus-ended. But remember that this is from your 101 biology, that during the metaphase of your cell division, all the chromosomes in double lines up in the equator right here. And then when anaphase starts, again, they are pulled towards each pole. And during this pulling, you need the destruction, or not the destruction, the depolymerization at the plus end. So the microtubule shrinks from the plus end, and that causes the chromosomes to be pulled towards the pole. So each of these chromosomes goes toward this, and then the cell will divide. So this is enhanced by this kinesin 13 and the stathmin, and we're going to talk about this much more detail. Remember that we completely omitted the 18.6 section of this chapter, which we'll talk during the cell cycle. Yeah?
How does it stay attached then?
Oh, that's a good question. So the complex, it is, it is really mind-boggling. So far, in between the kinetochore and the plus end of the assembly protein, there are more than 200 protein discovered, which makes that complexes. So it is an extremely complex process, and we're going to talk about that it disassemble, but there are still some of this. So basically, what happens is that it's an interesting process. You have this blunt end, and here you have the kinetochore of, and it attaches like this, and the motor proteins walk this way, and it disassembles this from here. Does it make sense? So it disassembles from here, but the motor protein walks this way, pulling those. So that's the process. We're going to talk about that when we discuss about the cell cycle. So yeah, very intelligent system. I mean, like you really it is. And so here now we are talking about the motor proteins. Now you may see that with the part 3 of 2.2, I posted a whole bunch of videos. Some of them are 4 or 5 minutes. Okay, all right. They will make you laugh, some of them. But those are your study breaks you can do. But at the end, the funny part is that at the end, there are some long videos, like 35, probably almost close to an hour. And the reason I posted that, and again, it is not necessary to watch those videos for your exam. It is only if you're interested, because I think that particularly is fascinating. So it is so I'm just distracting a little bit. So those videos are from Ron Vale. So he is a professor at the University of California, San Francisco, for a long time working in there. And he discovered kinesin. And then he also elucidated several structures of the dynein, although dynein he didn't discover. So he is kind of a godfather of these motor proteins. So what he did, that in 2006, he founded this iBiology, which is basically the discoverer of the scientist which discovered these phenomenal thing. They do have these videos available, so you can just search iBiology in YouTube and you will find that. So those two videos actually tells the discovery of the kinesin and then the dynein as well in detail. It's like a really complex. It goes into the structural biology as well. But if you're interested, you can. It is really not at the research level in the sense that it's not a typical research seminar. It is made for a kind of a general audience to give you an idea, and you can think about that. It's very interesting as something different I'm talking about. So back in India, he actually started a single molecule microscopy course with the Bangalore Institute of Science, and I went to this workshop in 1996. So when I was a graduate student at that time, so at that time I had a chance to interact with him for 5 days. It was a 1-week workshop. So he's an amazing guy. So you can take a look at those videos. So let's take a look at the real thing now. So here you have the plus and the minus end of the microtubule. And this kinesin is first group of motors that move towards the plus end. On the other hand, the dyneins are the second group of motors which moves towards the minus end. So remember that it's similar to the myosins in a sense that all myosins moves towards the plus end and only the myosin 6 goes towards the minus end, and you have the same thing in there. The only difference is that these proteins are much, much larger compared to your myosins. So for an example, somebody told that, think about that when you have to walk on the actin microfilament, which are much smaller, you need a small car or a scooter or a motorcycle to go into the alleys. Whereas when you go through the highway, you need a bigger motor, a bigger car, and it's kind of like an analogy. I believe that it is probably true. So here, You do not have to memorize this chart at all. This tells you that what are the different types of microtubule associated motors are available. These are cytosolic motors, mitotic motors. We are not going to talk about any of them. Those are the ones that I talked about. These mitotic motors are responsible for moving the chromosomes during the cell division. And then you have the axonal motors as well. So we are going to look at the cytosolic kinesin, cytosolic dynein, plus end goes towards plus end, goes towards minus end, and also the axonal dynein as well. So these are some of the ones that we are going to take a look and what they are. So the first, we are looking at the kinesin, very similar to your the mitotic, like your myosin. So you can easily see that you have a globular head and two heavy chains with a tail region, coil, and then you have a light chain, and this end binds to the transport vesicle. And the heads, these works with the ATP hydrolysis. These are the foot. You can think about that. These are the two legs on which they do walk on the microtubules, and that requires the ATP hydrolysis. So very generalized structure, similar to myosin. So here we are looking at some of the different types of kinesins, and what are these? So first we are looking at this kinesin 1, and this is a very common one, and we will mainly consider about these one. So you have these two heads that's attached to the microtubule, and on towards this end on the other side with an adapter, it is bound to an organelle, or it could be a vesicle. Many different things can be attached to that end. And also the other one is a kinesin 2. Here is a heterotrimeric, so you are, other than just a dimer of the same protein, they are different. You have 1, 2, and 3 chains, and they are different. All of them are different in the sense. So those are also in the same group. They all are transport organelle as well as vesicles from minus end to the plus end. Then you have another group, which is the kinesin 5. It's very different from these. So this is bipolar in the sense that at each end it does have the foot. So you have on this end you have a foot, you have this end you have the foot. So it binds two adjacent microtubules, and they make these microtubules reorganize in different areas. So these are very important for not only during the cell division, also to keep the proper microtubule or maintaining, think about that, maintaining the roads or shifting these microtubules in correspond to each other. These are responsible for that. These doesn't carry cargo. These are just only for the organization. And then you have the kinesin 13 that we talked about. It is, although it is called kinesin 13, it doesn't have any motor activity that is responsible for the disassembly of the ends. We talked about it's the plus end disassembly, but it can do the minus end as well. But the plus end disassembly is more important because it's very important during the cell division. So after that, we are looking at that how this transport happens, and this is the same way. This is the hand-over-hand movement that we talked about. We looked at some of the videos, and if we have time, we will look at a couple of more. And with the help of the ATP hydrolysis, they will move the vesicle and towards the plus end. So very straightforward. I mean, like at this level. So here we are looking at the motor activity of the cytosolic kinesin. This is also similar to the myosin cycle. Now, this is also another important thing. Just make sure that, please do remember that at each step, how the myosins are working. So these are very common questions, and it will be on your test. That say, for an example, the cycle can start at any point. So think about that. Your myosin is bound to the actin, where you do not have any nucleotide. The tightly bound, ATP comes in, it detaches, hydrolyzed to ADP and PI. It goes, swings the other area and touches. At that time, ADP is first released, then PI is released, and it just flexes at that time so that the actin moves. So make sure that 1, 2, 3, 4, you have those. So we'll have all the events lined up, and you have to rearrange them. So this is a typical question that is asked for these. So here what you are looking at, that you have this motor head, and again, as I said, that is hand-over-hand movement. You have the motor head right here, and the back one is bound to the ADP. So it hydrolyzed already the ATP, but it's still bound to the ADP. So now what is happening at the leading head at that time? So you can easily see right here, this the ATP is coming in, is binding to that at that step. On the other hand, at that point, see that as the your ADP is released, it will swing the backward foot towards the forward part. So you can easily see that will come towards from the back, this one right here, towards the front part. The linker is red. And at that time, you will see that the leading head will release the ADP at that time, and this will cause the empty side, and this is a tight bound form. And next, an ATP need to come over and bind to that so that it lifts up and ready to go, or it can change. On the other hand, you can see that now it released the PI. Once it released the PI, now it is again going back to this condition where it can swing back forward. So the process is again exactly the similar way that it moves for the myosin. And make sure that for the myosin also look at the animation. That will make it much more clear that what are the steps involved. So next we are going into the cytosolic dynein, and it's kind of a really amazing. This protein is a 1.5 megadalton. So that is the molecular weight of the whole complex is 1.5 million dalton. So you can just well imagine that when you to look at a certain protein, you call it a 30 kilodalton protein, 50 kilodalton protein, 100, 200, this is 1.5. So you can easily imagine that how big this complex is. And you can see it in the electron microscope, cryo-electron microscope. I just put some of these original videos from the discoverers as well. So what they have, it is a dimer, and it does have a region which we call as this stalk, and then you have a head domain, and these are the microtubule binding domain. So I'm just going from the smaller one later to the bigger one. So these are the 2 foot which binds to the microtubule. Then you have a stalk, and this stalk binds to this circle-like structure, which are made up of 6, which we called as a AAA ATPase. So these AAA ATPases are the ATPase associated with several activities. That's the reason called AAA ATPase. It's a typical protein that does many different functions in the cell, and it is a superfamily of proteins. So that's the reason it's called a AAA ATPase. But it does have 6 individual subunits. And then you have a dimerization domain right here in the middle, and then you have the stem region, and the other end binds to this dynactin right here. So the dynactin is basically, we're going to go into that in the next slide. This end finally binds to the cargo. So this is the cargo binding end through this dynactin complex. So if you are looking at that, you are looking at this stalk and the microtubule binding domain. This is the other dimer with the stalk and the microtubule binding domain. And then you have the central ATPase, and this is like a wheel which will rotate and change the conformation so that there is a movement. But the movement is very, very different compared to the kinesin. Kinesin is hand over hand. For dynein, that's the reason that it is so complex that I did not go into that. You can see the supplementary video, and there we can, I will talk about that, but you can look at that. It is much more complex. And then you have these linker regions which are connected. It will go all the way up, and it will be connected to the cargo. So let's take a look here. And here what we are seeing, that we just looked at this part. This is the dimer of the dynein, and from these structure, which are the linkers, it is attached to this complex which is known as the dynactin. And again, there are 8 actin molecules, ARP1, CAPZ, and you do not need to remember any of these constituent proteins. All remember that the important one is a dynactin complex, and this dynactin complex with an adapter is attached to the cargo. That's number one. That's the only thing. So again, I'm repeating what you need, that you have a complex dynactin complex, and the dynein motor is attached with the linker with that, and with the help of an adapter, it attaches with a cargo. That's your just one statement. The next one is that this dynactin complex is associated with another protein which was known as a dynamitin. It's a funny name, and we'll talk about that, why it is called so. And this dynamitin is basically uses another stalk-like protein, which is known as 150 glued towards the back, and it attaches to the microtubule as well. So just think about that this is a walking end. This is a walking end. This will walk over the microtubule towards the minus end. And here you have a trailing end to keep this whole complex together. And just think about that. This was from somebody's talk that I'm just stealing the words. They said that think about that on the freeway sometimes you see that these huge truckloads carry these mobile homes. You have these safety cars at the back. Think about that this complex is so big and carries such a big cargo, you need this safety car in behind so that there is no obstruction of the pathway of that. And I think that's very, very true in that complex, that how complex is that whole process is. So how it moves, and again, we were not going into the detail of the movement at all. So the only thing what happens, think about that whole complex, and the cargo is at the top right here. And this whole complex was right here in the gray area. And due to the ATP hydrolysis, you have a rotation of this central wheel. And when this central wheel rotates, it directly goes into an alpha helical rearrangement through this stalk and make a conformational change and changes the affinity of this head, which is binding to the microtubule. Again, that is not at all needed. And once that is happening, that it will lift up and go towards the front. Remember that we are only seeing at one side. You have two of them. Now the important part is that why we're not going into the detail of the movement. Here the movement could be many different types. So this is also funny. Think about this. You have a large load which is attached with a rope, two ropes. You are holding with that. So when you are pulling it, it's really heavy. You can do it in many different ways. So you can walk like this, hand over hand, just like kinesin. You can just walk like that. Or you can just walk like the inchworm as well. It does exactly all of those movements. It just depends. So it can walk hand over hand. That's one of the movement. Or it can just go like this. And these are not the specific step. And sometimes you think that these tiny molecular nanometer-sized motors, I mean, like how you really look at these structures. So see those videos, and it will make you kind of understand that these are not like a story. These are reality, that how they move as such. So this is basically the overview of that. The only take-home message is that the power of the ATP hydrolysis moves these material. That's all you need. Detailed mechanism not required. So here we are now looking at both of them, the kinesin and the dynein. So here you have the microtubular organizing center at the center of the cell, and all these microtubules, in reality there are many more, goes towards the periphery. And what you are seeing, that you have the kinesin and the dynein, both the motors are present on these microtubules. And let's take a look here. So here you have an endoplasmic reticulum, and here this is your kinesin. So it is trying to go towards the plus end. And all these stacks of the endoplasmic reticulum are arranged throughout the cell using this. On the other hand, you can have a retrograde transport with this dynein motor as well. So here the Golgi stacks are moved by the dynein motors towards the minus end. Secretory vesicle is going towards the plus end. On the mitochondria, you can see that there are both of these motor proteins, and it can go in either direction. On the other hand, early endosome, if it's going towards the cell periphery, it is attached with the kinesin. On the other hand, if you look at these pigment granules, it can go both ways. And that's the overview of idea that depending upon the movement of the material or the cargo, it could be either dynein or the kinesin. So here is an interesting thing that you can just look it up. It is not possible to provide all the material. So you have seen these chameleons, that how quickly they change the color. You've seen that basically. So what basically they do, that they have these cells with this chromophore with these pigments, and at one point of time, these pigments are towards the cell periphery, and suppose you are seeing the green pigment on the cell periphery, and it needs to change to pink, and all these green pigments are with the dynein motors, they are immediately goes towards the center of the cell, and the pink pigments are carried by the kinesin towards the periphery of the cell. And think about that, how quickly this can happen to the cell to change these colors. And this is done by these motor proteins. All right. So here, on the other hand, also another important thing, that the microtubules and the actin do crosstalk. So you get out of your freeway and then you can travel in a smaller roadside as well. So kinesin can carry this vesicle, and then it can hand it over towards the myosin, and it can move over the actin depending upon which direction it's going. So it is like literally now you can understand that if you look at that funny the supplementary video, that it tells you that exactly these are the facts, that the kinesin can be and actin interchange can happen as well for the cargo transport. So next, so that's pretty much all about your cytosolic kinesin and the cytosolic dynein. Now we are looking at these exonymal dynein, and they are present in the flagella, 1-2 per cell. Like 1-2 per cell means the human cells, only the sperm cells have just only one. But there are many flagellates which can have multiple of them, not even 1-2. Say Giardia have probably 4 of them. But many of the Euglena have a couple of them. So these are all protist and single cells. So other than that, in a higher eukaryote, only the sperm cells have that. So then cilia can be many per cell depending upon where it is. Say your lower respiratory tract, you have a large number of cells, pseudostratified epithelium contains a large number of ciliated cells. In the fallopian tube, uterine tube, you have large number of ciliated cells to move the oocyte. If you are a physiology people, there are some cells called ependymal cells, which also do have the cilia to move the cerebrospinal fluid from your brain ventricles. So there are many other regions where you have many different per cell, but there are also a lot of cells which does have just an antenna-like cilia. These are much less in number. So let's take a look at that, what happens. The important difference is that here the flagella and cilia, both of them are movable. They move due to a particular region. So locomotion, single cell eukaryotes, moving particles or fluid over the cell surface. Just one example I gave, tracheal cells, but this is what it is. So here what you are looking at is a sperm cell, and what is the important thing, that this is a single fluorescent tracking material. So the dyneins are attached, so next to each other you can see that there are 2 points, and over the time you can see that how this dynein movement causes a wave-like formation, and this wave basically propels the sperm forward. So how this can happen due to these dynein motors, that's what we are going to take a look at that. So the first, what we are looking at, that this is basically the cross-section of an exonyme or a flagella, both the same thing. So what you are seeing, that there are outer circle have 9 of these doublets, and you have an A ring and a B ring, and we know that arrangement is 13 plus 10, A and B rings. Then what you have, that in the very middle, in the central, you have 2 individual rings with all 13 structures, but same thing. They are not overlapping. You have a connection interbridge in between them. Then you have a sheath. So let's take a look at what are the important structure which are associated with this doublet. And remember that this is commonly known as the 9+2 arrangement, because you have this 9 outer ring and then you have 2 inner ring. And this is the electron micrograph. So what you are seeing, that where is the A ring and the B ring, make sure that you get this one. So the A ring is where you have the 13, and this is the yellowish, light yellowish color. Whereas the B ring is a little bit darker with 10 rings. So that's your A and a B. So now with the A ring, you can see that these are some of the proteins with some stalks and heads sticking out. These are the dyneins. These are the dyneins on each side that you are seeing. And these dynein from this A subunit will interact with the B subunit of the adjacent one. And you're going to see that in the next picture. Also, another thing that what you are seeing, that there is this purple or pink colored structure in between these doublets. This is a protein called nexin. And these keep all these 9 doublets in proper place. And also there are some spoke proteins we call spoke head and spoke proteins. These are also responsible for keeping all these 9 plus 2 doublet in a position so that they are all fixed in a particular area. So you can easily see that it is pointed out here that right here, this is called the inner dynein. On the other hand, these are called the outer dynein. So these are the 2 dyneins. So let's take a look at a schematic that how it is. So here the dynein do have a base. On the other hand, these are the heads right here. So if we just go back to the previous picture, previous picture, see that there are these 3 heads right here. So those are the heads. So those were free and these were attached right here. And if you just look at that where they were attached, the base was attached to the A tubule. So that's the thing that you note. The base was attached to the A tubule and the heads were free. So that's one of the things. Now what you are looking at, this gives you a more clear idea. It's very clear. You have this base right here. This is a stable association with the A tubule that we saw. On the other hand, the head attaches to the adjacent B tubule, and this is basically with the ATP hydrolysis. This attachment is not permanent, so this will move. So let's go back to back in here so that you can put it in the perspective. So think about that. These free heads, with the help of the ATP hydrolysis or it will bind to this B ring which is adjacent to it. This will bind to the B ring, and that's what we are seeing in this structure, to the adjacent B tubule. Now, if we look at this structure, this is the important part. So first, what we are looking at, this is the A tubule, just only one part. So think about that we are looking at a tubule here, then you have a B tubule here, and we are just looking at just this part in between them. Just a small slice of the whole circle. That's what we are looking at. So you can see the base is attached permanently to the A tubule. And at the top, this is the head that is being attached to the B tubule. And look at the direction of the polarity. You have the plus end on the right-hand side and the minus end towards the minus side. So when you have the activation of the dynein, with the help of a stimulus and with the hydrolysis of ATP, what it is doing, see that it is trying to move from this area towards the front, from this towards the front, here towards the front. It's trying to walk. Again, remember that it's trying to walk. You cannot completely walk past a certain point. The reason is that all these A and the B tubule are attached with this Nexin linker, so compared to each one of them, they cannot slide past or move. This dynein is not powerful enough to move them because they are attached. So instead of moving them or sliding them, which easily could have been done if they were not attached, they are bending these rings. And this bend, think about that, this is being translated by the whole ring into that wave-like fashion. And that's the way that the waveform translate into the flagellary and the ciliary movement. And how you know that this is exactly this bending is happening? You can use, right here, you have the green protein, which is the Nexin. You can use a protease and you can remove these linkers. At that time, you activate the dynein, and the A and the B tubule will slide past each other. So you have the flagella, it will be straight. There will be no movement as such because there is no bending of the A and the B tubule. So you can easily, using this experiment by removing the Nexin, you can just prove that the bending of the A and the B tubules by the dynein does that. So this is basically the role of dynein. Now this is another interesting part, and this is an entirely different area. So remember that here we talked about the bending of the flagella cilia for the motion. On the other hand, here we are looking at the transport, not the motion. And here we call this as an intraflagellar transport. So here you have the microtubules plus end is towards the tip of the flagella or cilia as well, and the base is towards the cell body. So what you are seeing that here, the blue ones are the kinesin motors. They are taking some of the cargoes towards the tip, and here there are some dynein motors. They are taking these cargoes towards the cell body. So think about that, that if you have some of these cilia which does have any particular receptor, those receptors are delivered by the kinesin, and something binds to the receptor, it is endocytose, and this receptor and the cargo molecule can be moved towards the cell body by the dynein. So this movement happens very frequently, and we'll talk about that. So now we are looking at a different type of cilia which is not responsible for moving fluid on the cell surface. This is present in many of the sensory organelles, mainly olfaction, odor, photoreception in your retina, in your eye, flow, kidney cells to sense the flow of the urine, many other tissues as well, present in most of the vertebrate cells, in the inner lining of your blood vessels to see the blood flow. And these are highly stable microtubule structure, and this is resistant to colchicine as well. And these are all non-motile. These lack the central microtubule. So the difference is that this is also a good quiz question. So remember that quiz question right here. You have the 9+2 arrangement where you have these central two tubules. These are the cilia and the flagella which moves. Movement is a function. Whereas for the sensory cilia, you do not have this central tubule. But you have the A and the B tubule. That's the same thing. You have 9 of them. And here is a picture, primary cilia on the kidney epithelial cell. And you can see that this is just like an antenna, just sticking out of the cell, just only very few. Sometimes could be multiple, but really not all over the cell. So what they actually do, and we will take a look at these. So here we are talking about 2 diseases which are responsible. One of these, and these diseases are called as a ciliopathies. This is the generic term of this disease. And one of them is a polycystic kidney disease. It's an autosomal dominant, and about 1 in 1,000 Americans are affected by that. So this happens due to the mutation of either 2 of these genes, and this is a polycystic kidney disease gene 1, PKD1. 86% of this, the PKD is due to that, and the rest is due to the PKD. So this PKD1 and PKD2 produce 2 proteins, and these are on the primary cilia. These are known as a polycystin 1 is produced by PKD1. Polycystin 2 or PC2 is produced by PKD2. Very straightforward. PKD1 is the gene. Polycystin is the corresponding protein. PKD2 is the gene. Polycystin 2 is the corresponding protein. So here you are seeing the green ones are the polycystin 1s, and associated with that is the polycystin 2. And here you are seeing some of the motors, dynein motors, carrying cargo, and absence of these can lead to this polycystic kidney disease, which can lead to failure. So basically what is happening, that the urine flow cannot be detected by these cells deficient in that. So due to that, there could be this cyst formation in the kidney and eventual kidney failure. So this is one of the disease. On the other hand, this is a very rare disease, and see that what happens here. This is known as the Bardet-Biedl syndrome. There are a couple of them in your book as well. Just I picked up these 2 are the important ones. And very rare, 1 in 140,000 live birth, and a lot. Obesity, polydactyly, multiple, more than 5 digits in each hand or feet, cognitive impairment, kidney cyst, really a large number. These are just a few of a large number of problems, but still this is compatible with life. I said that in the lecture video, that still the person can live with this disease. This is important. Retinal degeneration. So what happens that this is due to a mutation in one of the 14 genes that encode component of a particular protein, which is known as a BB-zome. So BB-zome is a protein, and it is very specific. See that we talked about the COPS and the clathrin, that they basically transport their cargo to a certain areas, COP1, COP2, then clathrin. Here, what BB-zome does, it gets attached to the specific receptors. So first let me tell that these receptors are present on the primary cilia. So think about that, just giving an example, that you have your olfactory cells in your nose and olfactory neuron itself. And then you have these primary cilia which are present on your dendrites, and those do have receptors for the odorant molecule. So you need that receptor to be present on that primary cilia to detect odor, and similar to many other. And these receptor molecules, these are all proteins synthesized in your ER and the Golgi, transported onto the plasma membrane. And once they are on the plasma membrane, BBsome binds to these specific receptor, and they take these to the primary cilium. So the function of BBsome is to transport the primary cilia receptor to the primary cilia. So in the absence of the BBsome, this receptor doesn't go into the primary cilia. So you will not be able to recognize an odor. On the other hand, think about that retinal degeneration, because you will not be able to detect light or those things, and it's a really debilitating disease. So this is the important part. So again, repeating that your BBsome is responsible for delivering cilia-associated receptors. In the absence of that, this disease, barred beetle syndrome, appears. So here is at the very last what I was saying, that the same author. So this picture is from the same group of author. This is a much earlier, whereas this is a same group of author in 2011 from the New Journal Journal of Medicine. So, and you can look at that this artwork is very, very cool. So right here you have the basal body, and see that these are in triplets, so these are perfect. On the other hand, if you think that this is a movable flagella, then you will have these doublets and the 9+2s, and see that what is present in here. You have the dynein in this purple color, kinesin in this blue color, and this is basically your anterogate transport. That means that cargo is going into the flagella or the cilia, and see that what is associated with the microtubule, the green proteins, these are the kinesin, moving towards the plus end. And here you have the PC1, PC2 complex that we just talked about. These are the receptors that is needed. On the other hand, think about that these receptors will receive some signals that will be a cargo, that could be endocytose, and now these cargoes are going towards the cell. And now see the switch. So you have the kinesin is, so let's go, move here one time before I go there. So these are the anterogate transport. You have the kinesin taking the cargo, and see that the dynein are hitching the right. This is the most important part. So what I want to emphasize here, that how does dynein go to the plus end if it is moving to the minus end? And how does kinesin go to the minus end if it has to only move to the plus end? So when a cargo is transported, they always hitch a ride with the other one. And that is the funniest part, that here the kinesin is carrying the cargo, and right here the dynein is hitching a ride to go towards the plus end. Now here the dynein is at work. It is now at retrograde. It comes towards the cell. Here the kinesin is hitching the ride to go towards the minus end. I mean, like, it's amazing that how these molecular motors work this way. As such. So that's pretty much will be the end of this chapter, and we are just looking at, just before we go into so I'm just showing you that where you find the videos, you can look at those videos. So you have the lecture videos right here. This is the part 3, and these are the funny videos. The first one you have seen. So this one is amazing, and it is very dramatic, but the fun part is that what is being said in here is true. I mean, like, so if you carefully look at that, so suppose a kinesin is moving, and what I'm saying, so right here, now see that how it is true. So how this is true, okay, so think about this. Okay, so right here your kinesin is going the hand-over-hand movement. That's a correct movement. On the side, this is a myosin which is hitching a ride. So if this needs to go from the microtubule to a actin or microfilament, this will help with that. On the back, there is someone else who was sleeping. That's your dynein. This is the dynein, which is basically hitching the ride in that process. And there's nothing to do because it will go to the plus end before that works. So on the other hand, the funny part that is being shown here, now see that this is an alley which is a microfilament. And now myosin wakes up and tries to go there. And it's a battle. If it can go, if it is not destined to go in there, it will not be able to. So it failed to go in there. So that's basically the process, that how it works. It's really funny. On the other hand, these are the animation out of a cryo-electron microscopic frames. So this is a microtubule where the heads are attached to that microtubule, and see the dynein motor is basically just hanging in there. So well, these are, I'm just trying to, so this is real picture. So this is how it's kind of they work. So take a look at those videos. Very interesting ideas. This is the Ron Vale, and let me stop these. And I will just show right here that how this movement of the at the very end. So let's go to the animation of that process. So see that how the movement is happening, and the vibration is just due to the Brownian motion. That's not ATP driven. So here what you are seeing, that these are the two parts which is attached with a high affinity, and this is the ATPase ring that is connected with the cargo. And these data are from molecular data. There are some of the areas that he will say that this area is grayed out because we do not know, we do not have a structural data. But what you are seeing is a structural data. So now you can see that it oh boy, sorry. Now it moved. So this is the, due to the ATP hydrolysis with the ATP, now it is now tightly bound. So this is the lagging head forward step. See that it is now trying to go. This is an inchworm kind of thing, that it is not passing the previous head that I was talking about. So it just binds right there. But at certain times, it can do this, and let's take a look at this. It can come back as well. So you can see that it's a back and forth movement. Now it passed. This is a hand over hand. So as I said, that it can either go this way, it can go back a little bit. And think about that. A person is trying to pull a cargo extremely heavy and trying all sorts of way to do that. And it's really amazing that how you can look at that. So these are from the structural data. These are all extracrystallographic data, worked his whole life in there. All right, first quiz question. A little bit from intermediate filaments are distinct from microtubules in all of the following excepts. I just particularly put it this way because a lot of people got confused by the wording of that, what to choose, so that you just make sure that you clearly understand that when I use an except, what we mean by this. All right, stopping it. Oh boy. Still, the highest 29% is the winner still. So that's a good sign. So intermediate filaments are more dynamic. They are not, because see that once they are formed, they are there in the place all the time. So they do not depolymerize and polymerize. Intermediate filaments are not directional. So intermediate filaments do not have associated motor proteins, correct. Intermediate filaments do not need ATP to assemble. and of course there is a difference. So the intermediate filaments are more dynamic. So that is wrong because so just make sure that this type of question could be asked. So let's go to the next one. So microtubules, microfilaments are similar. This is an easy question. Both use GTP as their energy source. Both have plus minus ends. Both can disassemble by catastrophe. Both assemble from a small number of locations in the cell, none of the above. Yeah, that's very correct. That's correct. Both have plus and minus end. Remember that. So one thing that just remember that both can disassemble by catastrophe, that doesn't happen. Remember that this kinesin 13 and the stathmin, the plus end binding proteins, they can disassemble at the plus end, and it is needed for the movement of the chromosome. And no such event happens. So the plus end, usually if the critical concentration, technically it can happen, disassembly, but actively the disassembly or the depolymerization of actin at the plus end doesn't happen. So that's why that's not the answer. All right, so the last one. I think this is also straightforward. Force for axoneme bending is derived from the bending of the outer doublet microtubule, bending of the central singlet microtubule, sliding of the central singlet microtubule, and sliding of the outer doublet microtubule. Make sure, stopping it now. Yeah, bending of the outer doublet microtubule. Now here, make sure that this question could be asked in a very specific way as well. So just remember that, I told you that you probably noted down, but make sure that when we're talking about the axonemal dynein, you know that where the base binds, A tubule or B tubule, where the head binds, A tubule or B tubule. This is a very common question, and it is very easy to mess up that. So base always binds to the A tubule, and the head binds to the B tubule, which tries to walk towards the minus end. So just make sure. And the easy way to remember that, I guess, that remember that picture of the whole 9+2 arrangement. See that it is attached with the base at the A tubule, and the heads are radiating out. So you know that that's the head which can bind and not a stable structure. All right, so let's go back to a little bit better. A little bit better. So again, this whole thing, I will upload this PowerPoint file as well. So, and you will have the recording. So if you just really cannot see from the back, just one time look at the PowerPoint and just watch this part if it is necessary. So let's take a look at that, what we are looking at. So overview of the, so the first thing that what I'm trying to say is that whatever is in here important, and why some of the areas are highlighted, because these are some of the area that I historically see that people struggle with in the exam, in the second exam, usually. So those are the areas highlighted. So rough purification of the ER signal hypothesis, of course, those are important, but this is the most important area, and that's why I posted long back, probably February 8th, that question in the discussion board, is the type 1, 2, 3, 4, 5, like 4A, 4B, and the GPI anchored proteins. So if you remember that, for each one of them, you have a particular topology, N and C. And I posted yesterday an announcement. There was a question which was in your practice quiz and the quiz. I removed that question, which says N in and C out. Do not worry about that. If I just use the word, I will say N in the cytosolic side, C on the lumen side or something. It will be specifically written. So that's not a problem. But one of the things that most of the people struggle with, remember that when you have these particular sequences and what they do mean, you will have two different types of sequences. One is a signal anchor sequence, and then you will have an STA, which is a stop transfer sequence. So mainly these are the two things that is very important as such. So what these different things means. One thing where people get confused mainly, suppose if I just write this one N and the C terminal, that in the mechanism that when you have the ER membrane and the translocon is sitting right here, most of the time the start picture is showing ribosome sitting in here, and the protein is coming out from the ribosome. Either it is coming into the lumen or it is on this side, something like this. Remember that if you do not have a signal sequence, again, I'm repeating, if you do not have a signal sequence which is at the N terminal, until the first SA sequence or the STA sequence appears, the synthesis still goes on in the cytosol. Because remember that this is hydrophobic. This is hydrophobic. But in the N terminal, this part is not hydrophobic. So you have the ribosome here, and there is no problem of this N terminal part to synthesize there. So why that signal is important? So think about this, that you have the ribosome right there, and you have this part coming out. Now, right here you have the hydrophobic part appear, which could be either a signal anchor sequence or could be a stop transfer signal, depending upon what the condition is. As soon as that appears, this is not compatible with the cytosol due to the hydrophobicity. And immediately an SRP or the signal recognition particle will come in and bind. It will not bind until you have an SA or STA sequence comes out, because this part is completely polar and compatible. And then it brings the ribosome at the translocon. So other than the signal sequence, and this part is not shown because you see the first picture is the ribosome sitting on the translocon. But remember that before that, a small portion happened. And that is this part before an SA sequence, or if it is an STA sequence, that's fine as well, that this portion is synthesized in the cytosol, and once this appears, then only the SRP brings it towards the translocon. So just make sure that you have that. That's one of the things. Another thing is that if you see that you have the plus charge amino acid on one side, remember that will be always towards the cytosolic side. Cytosolic, this is the lumen. So if you have a protein like this, if you have a plus charge, you will have that on this side. So that's very important. And again, we cannot just talk about just only this thing as such, but the basic difference between the two is why you have the stop transfer and the other one is the signal anchor sequence. So what happens, your signal anchor sequence is appearing, it's a hydrophobic part, and once the signal anchor sequence comes in, it goes to the translocon. From the lumen, it has to diffuse into the translocon. From there, it has to diffuse in the membrane. So that's the time required. Whereas for the stop transfer, what happens, that the time it takes to diffuse out in the membrane, that is when the ribosome is not making the protein and it's stopping. Once it's out from the translocon into the membrane, then the rest part is synthesized. So just make sure that this part, I will draw it down and spend a good amount of time, and if possible, just work with your friend or in a group study to do that. This is one of the area that even the graduate classes, they struggle with these questions. So then remember the process here, the processing of N-linked oligosaccharide in the ER and the Golgi. And remember that when you're reading the question, make sure that many of these alpha glucosidases and mannosidases, the names are same in the ER and the Golgi. So pay attention whether it is written as ER glucosidase or Golgi glucosidase. That could be the thing, that just that small word can have an entirely different meaning. So make sure that choose those answer, that you have to look at the prefix before the glucosidase or mannosidases in there. Another important thing that a couple of you guys asked that if you remember that there is a whole series of synthesis of the biochemical reaction for your the glycan synthesis or N-acetylglucosamine synthesis along with the mannose. Do you need the steps? Steps are not required. So what is the take-home message and what is required? Remember that at one end you have a dolichol phosphate, and after a couple of steps, addition of couple of sugars, it flips inside. It flips inside with all these sugars. Still, it gets addition of other more couple of sugars. Once that is complete, this whole structure is transferred to the protein. So if you remember that whole process, that the dolichol phosphate is the lipid which is responsible for making it, first it is towards the cytosolic end. After addition of some, it flips, then some addition, then it transfers the whole thing. And also remember the asparagine X serine threonine, that sequence is responsible just like a Cadel sequences. Then also this will be your question from your endo H and endo D experiments, and follow the PowerPoint slide that when it passes the medial Golgi and what happens. So this will be always there. You will have a picture or a schematic picture of an SDS page, higher molecular weight or lower molecular weight, and make sure that you completely understand that's what's going on in here. So I'm just giving you a couple of examples that if you just have a panel like this, you have a 10, 15, 20, 25 minutes. And if you just do have something like this, you have something like this, and suppose this. So from here, I'm just asking you that this is your protein right here. For the first one is a control and then the experimental. And same thing, each one of them is a control and experimental. So if you are looking at that, from here, can you tell that which enzyme I looked at? This is basically a protein moving from the cis Golgi towards the trans Golgi, and I have used some of the enzymes. And again, this will be very specific. Whether it is endo H or endo D, can you tell? Who tells D? Yeah, that's correct. So remember that. Suppose if this is the medial Golgi, after it passes the Golgi, it becomes sensitive. That's why the molecular weight is low. This will be entirely opposite if it is like this. Before it passes the medial Golgi, now it becomes endo H. Before it passes, it can cleave. Once it passes, it cannot. So just make sure that when you read these experiments, you will have some critical thinking questions from here. So make sure you remember that. Additional info about that endo D, extra material, experimental protocol, not needed. Those are remember those mannose 6-phosphate hydrolases, M6P, those are important areas. All right, so right here. Here, this is important here. So fate of the endoglycosylated receptors, remember that your LDL and the transferrin receptors are recycled until there is always you can argue if the receptor goes bad at some point of time, it will of course go, but in almost all intent and purposes your LDL and transferrin are recycled. EGF is destroyed, whereas the transcytose is the Fc receptor for the which binds to the Ig's Fc area. So remember that. This is a very important part. Remember the clathrin and the COP1-2 and the snares, RAB-GTPase. This is a complex area. So make sure that you really know the step-by-step paths with that. Know the critical concentration. So remember that you will not have to calculate anything. But remember that I tell you a critical concentration right here. You have a minus C minus and you have a CC plus. I give you some of the concentration right here. Say, suppose here I give you 10 micromolar. Here I'm giving you the concentration of 5 micromolar. And I'm saying that at this, you have the concentration of your, in the cell, you have a 3 micromolar concentration. At this concentration, what you are going to see? You're going to see is addition on this end or that end. Does it make sense? What do you think it could be? So I mean, like, try to think about this question, that sometimes at this point it is still closer to that, and maybe at some point of time it can add it. But if I just, instead of that, if I say 7 micromolar, then what happens? That adds to what end? Plus end. So that's what it is. So if you have these concentration, that at what concentration it will specifically add to that minus, and remember that it has to be less than this but more than this. If that happens, it will add to that end. So these are just a basic idea that if you have the numbers, remember that you do not have to calculate though anything. And these are a lot of different proteins, so it needs a good amount of time. The actin binding protein, motor protein, and the motor protein step size, how you can identify those. Those are important, and the neck length is related to the step size. You can find some of the questions which could be, you will given a chart just like the step size, and you have to look at the diagram of a couple of different myosins and say that whose step size is small, whose step size is big. So you may have to identify those. And the very last part, I guess, that we talked about these major classes and distribution example for the intermediate filament, and the kinesin and dynein, just to make sure that you remember the plus minus end and the microtubule and the axoneme. So that's about it. I'm just putting the post-it. All right, good luck.